Hi everyone, this is lecture 3 on synaptic transmission. In the last lecture we learned about the electrical events that occurs in a neuron and how an action potential is produced and propagated. Now we're going to look at different ways synaptic transmission occurs in neurons. And you already have an idea from SCB 203 when he learned about the neuromuscular junction. That was an example of synaptic transmission. So if you understood that, then the rest of this should not be too much of a problem. So let's begin. So essentially we're going to talk about the events involved in synaptic conduction. How are neurotransmitters categorized? Okay, you know one already, acetylcholine. What are the effects of a neurotransmitter on the membrane potential? And what are the mechanisms to remove neurotransmitters from the synaptic cleft? Because it's one thing to be able to start synaptic transmission, but you would like to also stop it. Okay, so let's begin at the basics. A neuronal synapse. What is it? Well, it's a structure that neurons use to communicate with other neurons. Okay, it doesn't have to be another neuron. It could be a muscle cell. It could be an endocrine cell. Okay, so it doesn't have to be a neuron. There are different types of neuronal synapses, okay? And the differences between them are which structures are forming the synapse. So we have axodendritic, axosomatic, and axoaxonic. I think you can get a pretty good idea of what each of these are. So an axodendritic one is where there is a synapse between the axon of one neuron with a dendrite of another. Axosomatic is from the axon of one neuron and the cell body of another. And finally, axoaxonic is a neural synapse between two axons. Okay, so let's go through these anyway to make sure everyone is up to speed. Axodendritic synapses. These are neuronal synapses between an axon and a dendrite. Okay, so here I circled here the axodendritic synapses, connection between an axon and a dendrite. So we hear the synaptic bulbs or synaptic ends of one axon forming a synapse, the dendrites of another neuron. The second type are axosomatic synapses, a synapse between an axon and a cell body. Here I circled this one. So here it's showing an axosomatic synapse, a connection between an axon and the cell body of another neuron. Finally, we have axoaxonic synapses, synapses between an axon and another axon. Okay, so here's a synapse between the axon of one neuron with the axon of another neuron. So these are not too terribly complicated. You just got to remember what each of these prefixes and suffixes mean. Okay, we have terms now that describe neurons that send and receive messages. And these terms are presynaptic neurons and postsynaptic neurons. Okay, I think you could get a pretty good guess at which one is which. Okay, so here's a very simple cartoon from your textbook showing presynaptic neurons and postsynaptic neurons. So in blue is the presynaptic neuron, in pink is the postsynaptic neuron. The presynaptic neuron is sending the message from its axon. So we're forming a synapse here, okay? And the presynaptic neuron is sending its message to the postsynaptic neuron. Likewise, the postsynaptic neuron, which is pink, is receiving the message from its dendrite, cell body, or axon from the presynaptic neuron. Okay? So presynaptic neurons are sending the message, postsynaptic neurons are receiving the message. So, I mentioned this several times already. So do you remember what type of synapse did you learn in SCB 203? Well, that was the neuromuscular junction. There's a synapse between the axon of a neuron and a skeletal muscle cell. Okay. So let's look at the differences and compare the characteristics between neuronal synapses and neuromuscular junctions. Since you know kind of a good idea what neuromuscular junctions are, how do neuronal synapses appear differently? So here I made a little table showing the major differences. Okay. In the neuromuscular junction, the muscle fiber receives input from a single axon. So you have one axon, one muscle fiber. However, in neuronal synapses, neurons may receive input from thousands of axons, and this might be even an underestimate, meaning showing how complicated the circuitry is in the nervous system, making it very difficult to map and to understand the kind of wiring or blueprint that allows our nervous system to function. Another difference is that in the neuromuscular junction, you have one choice, and that's acetylcholine as the neurotransmitter that excites the muscle fiber. Okay, so this was actually pretty convenient for SCB203 because you didn't have to worry about which neurotransmitter. You only had one. Okay, but in neuronal synapses, it's much more complicated because you have a variety of neurotransmitters that may excite or inhibit the postsynaptic neuron. Okay, so in the neuromuscular junction, acetylcholine excited the muscle fiber, but in neuronal synapses, you may have different neurotransmitters that excite or inhibit the postsynaptic neuron. Finally, the action potential in the neuron triggers an action potential in the muscle fiber. Okay, we saw that action potential from the neuron stimulate the neural muscular junction to release acetylcholine, and eventually that would cause an action potential of the muscle fiber. Muscle contraction was going to occur. In neuronal synapses, like in the first one, a postsynaptic neuron may require input of more than 100 or more presynaptic neurons. Again, this is probably an underestimate. Okay, so now we know what a synapse is and the different types of synapses. Let's look at what synaptic transmission occurs. Synaptic transmission is the transfer of chemical or electrical signals between neurons at a synapse. Okay, so the key words here is the transfer of chemical or electrical signals. Okay, we're pretty used to thinking of chemical like in acetylcholine, but you can also have electrical signals at a synapse. Okay, so that means there are two different types of neuronal synapses. They may be electrical or they may be chemical. Let's begin with the lesser known of the two, and these are electrical synapses. So these are important for programmed behavior such as breathing, because breathing you want it to be, you don't have to think about it too much, it's automatic, and you want it to be quick. And so what better way than to have electrical synapses, because whenever you introduce a chemical, that's going to slow down the process. So electrical synapses should be quicker. Okay, and we see that neural synapses are only separated by 3.5 nanometers. They're very close together, and they're connected by gap junctions. And you know about gap junctions from studying the heart in SCB203, because the gap junctions allows the heart to act like a syncytium. A syncytium is a group of cells that share the cytoplasm. It's not a real syncytium, but it's a functional syncytium, because since the cytoplasm can pass through the gap junctions, depolarization of one heart cell will depolarize the other heart cells. So the heart will excite and contract as a unit. By the same token here, these gap junctions here will allow free movement of cytoplasm, so that when the presynaptic neuron is excited, it will automatically excite the postsynaptic neuron. And so we can hear in this inset here, we can see the gap junctions with the ions passing through them, through the gap junction channels. So the gap junctions allow electrical current to flow from one neuron to the next, similar to that of the heart. Now, another unique feature about electrical synapses is they're bidirectional, meaning they can go both ways. It could go from one here, the first axon terminal to the second one are backwards. Okay, and as you might expect, as something that's electrical and so close together in distance, it's going to be nearly instantaneous. There's only like a 1/10 of a millisecond delay. Okay, and then of course, something like breathing is something you'd want to have something not to be delayed too much. Now let's spend more time talking about the other type of synapse, which are called chemical synapses. And these are the majority of synapses in the nervous system. Okay? Chemical synapses are more efficient and unidirectional, meaning they only go one way, from the presynaptic neuron to the postsynaptic neuron. The electrical signal is converted into a controlled chemical signal with no loss of strength. Okay, so you might say, well, why would a chemical signal be more efficient? Well, by introducing a chemical, we have a way now of controlling whether we want it to occur or not. Sometimes we may want a synaptic transmission to occur, maybe we don't. It allows us the flexibility of having multiple mechanisms of action, especially when you have many different neurons synapsing on a single postsynaptic neuron. As you expect, it's going to be slower. So there's a chemical synapses are slower with a thought of 5/10 of a millisecond to synaptic delay. So this time it takes is called synaptic delay, and as you might expect, it's due to the time it takes for the chemical or neurotransmitter to diffuse across to the postsynaptic cell. Okay. And chemical synapses are just like the neuromuscular junction, which is an example of a chemical synapse. The presynaptic neuron has synaptic vesicles with neurotransmitters. Okay, so in the neuromuscular junction it was acetylcholine, but we know already that in neuronal synapses you might have a variety of possible neurotransmitters. Synapse is separated by a 20 to 50 nanometer cleft, synaptic cleft, so it's a little bit further, and it takes a while for the neurotransmitter, once released by exocytosis, to diffuse across to bind to its receptors on the postsynaptic neuron. And I got ahead of myself here. The postsynaptic neuron has receptors for the neurotransmitters. In the neuromuscular junction, it was the acetylcholine receptor. Okay, and many of these act as channels. Okay, again, I got ahead of myself here. Receptors are usually linked directly or indirectly to ion channels. Okay, what do we mean by directly or indirectly? Directly means that the channel is part of the receptor itself, like the acetylcholine receptor. Indirectly means that the channel is separate from the receptor. Okay, and the signals can vary in size. Let's look at the events at a chemical synapse then. Okay, and these are very similar to what you learned for the neuromuscular junction. First step, an action potential in a presynaptic neuron triggers calcium channels in the axon terminals. Okay, in the last lecture we learned how an action potential is produced as it propagates along the axon in the presynaptic neuron. When it gets to the axon terminals, voltage-gated calcium channels open in the axon terminals. Okay, so here's the action potential coming down the axon. As it depolarizes the synaptic terminal, these voltage-gated calcium channels open up, and so there's more calcium outside the cell than inside the cell. Calcium will diffuse into the synaptic terminals. Okay. Now, what happens to the calcium that enters the cell? Well, what it does is it causes the release of neurotransmitters into the synaptic cleft. What it causes is a calcium-dependent exocytosis of neurotransmitters that are now released from the presynaptic terminal. We'll say it one more time. It's a calcium-dependent exocytosis of neurotransmitters. Okay? Now, the neurotransmitters that are released into the synaptic cleft diffuse across to the postsynaptic neuron and bind to receptors there. Okay? So here, the neurotransmitters diffuse across and bind to the receptors. Then ion channels open, leading to a local potential and possibly an action potential. Okay. In our example here, we're going to say it's going to open the channel. Okay. We're going to find later that it could also close or inhibit a channel. But for now, we'll keep it as open a channel to keep it simple. Okay. And to produce a local potential. Okay, we know about local potentials and possibly an action potential. It depends if the local potential is strong enough to reach threshold to start an action potential. Okay, so we started a synaptic, we know how we start a synaptic transmission, but how do we stop it? Because we don't want it to go on forever. And it must stop to allow for the response to be stimulated again. Okay, so there are three ways to stop synaptic transmission. One, all the neurotransmitter diffuses across and is absorbed, and absorption of the neurotransmitter. Okay, so here the neurotransmitter diffuses away. Okay, and eventually it gets absorbed by the neuron. The second way to stop synaptic transmission is by its degradation in the synaptic cleft. Oh, we know this at the neuromuscular junction because remember acetylcholinesterase? That enzyme was used to break down acetylcholine into acetate and choline molecules. Okay, so degradation, neurotransmitters are degraded by enzymatic reactions in the synaptic cleft. Okay, and then these can be reabsorbed as well to make new neurotransmitters. Finally, a third way is the reuptake into the presynaptic neuron. Okay, so how is this different from the other ones? Well, this one there's a specific uptake mechanism using facilitated transport to pump it back into the presynaptic neuron. So those are the three ways we can stop a synaptic transmission: by diffusion and absorption, degradation, and by reuptake. Okay, so now we know the different types of neural synapses. We know the structure. We know how synaptic transmission occurs. We know how we stop it. But now let's look at the different neurotransmitter receptors. Okay, we mentioned they can be directly or indirectly linked to a channel, and there are two types. There's ionotropic neurotransmitter receptors, and there are metabotropic neurotransmitter receptors. Okay, let's take ionotropic receptors first. As you might expect, it has the word ion in it. So these are receptors that are part of a ligand-gated ion channel. They have rapid but short-lived effects because as soon as the receptor binds, the neurotransmitter binds, it can open or close. Okay, so here's an example of an ionotropic receptor with its combined ion channel, and it's closed. Here, a neurotransmitter binds to the receptor, and now it opens. So this is an example of an ionotropic receptor. So there's very little delay time since the channel is built in already to the receptor. The second type of neurotransmitter receptor is called metabotropic receptors. These are receptors that interact with a separate ion channel. So the ion channel is a separate molecule. So the receptor has to somehow activate the channel. Now, most interact through intracellular enzymes called G proteins. Okay, so you might have heard of these in SV203, but you might not. It depends on if your lecturer went into that much detail. Most likely, your lecturer might have said it in passing, but didn't go too much detail. Okay. Now, G proteins are a special set of proteins which allow communication between receptors and ion channels. Okay, and these G proteins operate by producing what's called a second messenger. that opens or closes the channel. And the second messenger is produced by the G proteins that will amplify the signal. As a result, it's longer lasting and more variable. Okay, so here's an example of a metabotropic receptor here. Okay, here's the G protein here in green. Okay, the receptor is in orange. Of course, the ion channel and the metabotropic receptor is separate. From, the channel is separate from the receptor. So this is when it's inactive. Now, when it's activated, the neurotransmitter binds to the receptor. Okay, this stimulates the G protein to be activated. Okay, this causes a series of enzymatic reactions to produce a second messenger. Okay, now as a result of the second messenger, which is now amplified because it's not just 1-to-1, it may be 1 to 10,000. Each of these second messengers can now stimulate a different ion channel to open it, for example. Okay. Of course, it could close it as well. But in our example here, we're opening the ion channel. In this case, it's going to have a bigger effect because now it can possibly interact with many more channels. Okay, but it might take a little longer because of these additional steps that are required to activate the ion channel. Okay, so again, many metabotropic receptors they operate through G proteins that will activate the channel, which is now a separate molecule. Okay, now what's left in our discussion? Well, we haven't talked about the neurotransmitters. Okay, so let's talk a little bit about the major neurotransmitters. And we can classify them in two separate groups. We have excitatory neurotransmitters, and we have inhibitory neurotransmitters. We're going to eventually talk about their effects, and so I'm going to tell you to you now, but we might not describe these terms more until the next lecture. Excitatory neurotransmitters produce what are called EPSP. These are called in-plate synaptic potentials. Oh, sorry, excitatory postsynaptic potentials. Okay, let me say that again. Excitatory neurotransmitter produces excitatory postsynaptic potentials. Okay, inhibitory neurotransmitter produces inhibitory postsynaptic potentials. Okay, seems kind of redundant, but because we'll be using these terms frequently, and so we just want to make sure that you understand what they mean. Oh, okay. Okay, need to go to the next slide. EPSPs are excitatory postsynaptic potentials, and IPSPs are inhibitory postsynaptic potentials. Okay, so we'll just introduce this term, and we're going to come back to these in the next lecture, I think. So let's look at the types of neurotransmitters. We have 4 groups of different neurotransmitters, and you know the first one already. Well, because it only includes one neurotransmitter, and that's acetylcholine. Okay, so that's pretty easy. So the first group is easy because there's only one, and you know it already. The second group are called biogenic amines. They're also called monoamines. Third are called amino acid neurotransmitters, because some neurotransmitters are formed by amino acids or modified amino acids. And the final ones, some are proteins, and these are called neuropeptides. So these are the 4 different groups of neurotransmitters. Let's take the first one, acetylcholine. These are found in cholinergic synapses. Okay, so these are the ones that, of course, use acetylcholine. Now, this term cholinergic, we're going to, when we get to the autonomic nervous system, we're going to talk more about these type of receptors. For now, just humor me and say that they're found in cholinergic synapses. Okay, so they're found in the neuromuscular junction, brain, spinal cord, and the autonomic nervous system. Oh, okay. And most of these are excitatory. Okay, so the neuromuscular junction, you know it's excitatory because we get muscle contraction. And it's degraded by acetylcholinesterase into acetic acid and choline. Okay, I said acetate earlier, but that's just the salt form. So acetic acid and acetate, they're equivalently the same. The second group are called biogenic amines, or monoamines. And there are 3 different groups within these. These are catecholamine, serotonin, and histamine. The catecholamines include neurotransmitters that you heard of before: norepinephrine, epinephrine, you've probably heard of already. You might have heard dopamine just from your own reading. And dopamine is from an amino acid from tyrosine. And they may be excitatory or inhibitory. Okay, so I guess there's some crossover between these groups. Okay, so dopamine is a biogenic amine that happens to be from an amino acid. Tyrosine is one of the 20 amino acids. So it's a little confusing because these categories are not so clear-cut. Serotonin is another group, and again, this is from another amino acid called tryptophan. Okay, and it's involved in mood regulation. Oops. And the last one I actually skipped already. I think it was what, tryptophan? What was the third one? Okay. Neuropeptides are the final group, and these include some examples. There are many of these. Okay, for example, substance P, opioids, and neuropeptide Y. Substance P functions in pain and temperature. Opioids includes endorphins, enkephalins, dynorphins for pain relief, and are depressants. Okay. Neuropeptide Y is involved in feeding behaviors. Okay, so I might have skipped a slide. Oh well. Okay, finally, here's the table that's included in your textbook that summarizes different types of neurotransmitters. Okay. However, you'll only be tested on the ones that I included in the text slides. Okay, but this allows you to see them all at once. Okay, so this is the end of lecture 3. Okay,